I- mutation causes lac repressor to have its DNA binding domain mutated so that it can no longer ever bind DNA, and that's why transcription is always on, because repressor can never bind DNA. The OC mutation is a mutation to the operator, which is a DNA sequence upstream of the lac operon, and that makes the operator DNA sequence different so that repressor doesn't recognize the new DNA sequence, and this is why this is a constitutive mutation as well. That leaves C matching with 2. The superrepressor mutation of LAC-I, which again is lac repressor, changes the shape of the site where lactose binds lac repressor, and it makes lactose unable to bind lac repressor. And remember that that's the trigger that causes lac repressor to be removed from the operator. So in this case, lac repressor can never be removed from the operator, and that's why transcription never happens. Here the answers are 2 and 4. And that's because if A is present and there's no B, then A will prevent B from turning on, and so A is always present and B is always absent at low levels. And of course, these diagrams don't indicate the idea that the amount of protein A will slowly decrease over time, but even if it did, if there was no B to start with, there would never be an opportunity to produce B in this situation, at least until all of the A was gone. Option 4 works because there's a little bit of B present, and so it causes a slight reduction in the amount of the protein or transcript A that's produced. Likewise, because there's A present, that causes that drop in the amount of B, and then we return to a graph that looks very much like plot number 2, where there are 2 horizontal lines. In the absence of B, there's no change in the system. So we have the lac operator, and here we have the lac operon encoding the lac-Z, lac-Y, and lac-A genes. And in the absence of lactose, lac repressor, which is the product of the lac-I gene, whose transcription produces a messenger RNA molecule, and then translation of that messenger RNA produces the protein lac-R, lac repressor, which is also called lac-I. And that protein is bound to the operator in the absence of lactose. That means that the repression of lac-ZYA occurs, and you get no transcription. Normally, the purpose of lac regulation is to turn on a suite of genes, including the gene lac1. And this happens using this system where there's an upstream activating site, which is where the protein Gal4 binds as a dimer. And that protein complex will positively interact with proteins to recruit things like RNA polymerase to the transcription start site to transcribe lac1, enabling the yeast to use gal1, to use galactose that's present in the medium. But the protein gal80 binds to the activation domain here in gal4, and that prevents gal4 from turning on transcription. In the absence, therefore, of gal80, if there's no gal80 gene present and there can never be any gal80 protein produced, then you would predict that gal4 would always be recruiting RNA polymerase to the gal gene, so you'd get constitutive or always on expression of gal1. We've talked about a few different experimental approaches used to make GMOs. One is traditional breeding, that is waiting for nature to create mutations that cause phenotypes that we select for artificially by breeding organisms together. That's traditional breeding. Another example, this is not the only other technology we've talked about, was the use of the mitotic spindle poison, microtubule poison colchicine, which was used to produce seedless watermelons. Seedless because they're infertile. Two molecules that get epigenetic modifications are DNA and histones. And an example of each of these is we have CPGs, that's cytosine linked with a phosphate group as normal in DNA, next to a guanine. And in this case, the cytosine molecule is often methylated. So this is an instance where DNA itself, the cytosine, gets chemically modified by the addition of a methyl group. And methylation of DNA has an effect on chromatin structure and gene expression. Histones can be methylated, acetylated, and there are also other chemical modifications we haven't talked about to histones. And those are the proteins, the histones, that combine with DNA to create nucleosomes and form chromatin structure. So chemical modifications of those histone proteins by the addition of methyl groups and acetyl groups affects their structure, whether they're condensed or not. We don't expect to see male calico cats because cats are mammals, and like all mammals, males have an X chromosome and a Y chromosome. Well, I should say almost all mammals have an X and a Y chromosome for males and two X's for females, and it's only the XX sex that undergoes X chromosome inactivation. Because the male cat only has one X chromosome, that one allele, or one version of the X chromosome, has one allele of the pigmentation gene, whose alternative expression, based on whether or not one X chromosome or the other is turned off in females, affects the pigmentation. So if a male only has one allele and they don't do X chromosome inactivation, that should lead to a uniform or single coat color. So in the case where we did see a male tortoiseshell cat, we might predict that that would be a male because it's got a Y chromosome, and that it's tortoiseshell because it has two X chromosomes. And that would be akin to a human that has Klinefelter syndrome, two X's and a Y. So male appearance, but as we discussed in class, in Klinefelter patients, one X chromosome does become heterochromatinized and inactivated, turning into a very dense chromatin structure called a bar body. And if those two X chromosomes had different alleles, say one allele that produced orange pigment and one that produced black, then you'd expect to see a tortoiseshell cat. It's possible that one of the main genetic differences between adults that can't process lactose and those that can are that there's a mutation in an intron of a gene near the LCT gene, which encodes lactase, which is the enzyme that infants use and produce to break down lactose into sugars that we can metabolize. And this mutation in the intron of MCM8 seems to create an enhancer sequence, or at least improve an enhancer sequence, so that an enhancer protein will bind there in adults and positively interact with LCT, lactase, to continue its production into adulthood. If all I could see of this cat was its head, you might think that this X chromosome inactivation occurred at the 2-cell stage, where in one cell there was an X chromosome that produced an orange pigment and one that produced the black pigment had become inactivated, and that on the other side, the chromosome that carries the black pigmentation allele was active, and it was the red version that was inactive. However, because we can see some orange and some black down here on the tail, it's possible that, and there's no right answer to this question because we can't see the entire cat, it's possible that it was at something like the 8-cell stage where X chromosome inactivation occurred, such that perhaps one of those cells or a group of those cells produced the face, and some other cells that were inactivated produced that orange pigment that's present on the tail. So given the large blocks of color rather than little small spots of color, this at least seems like it was an X inactivation step that occurred early in development, 4-cell stage, 8-cell stage, maybe the 16-cell stage. But the fact that we can see 50-50 pigmentation patterns on the face suggests that it was rather early in development. The mutation will cause an actual change in the sequence of a DNA strand. For example, a mutation of T might replace a T with an A. This changes the DNA sequence. An epigenetic change does not create a mutation. It doesn't change the actual sequence of the DNA. So that's a main difference between mutation versus epigenetic change. However, as you know, chemical changes do occur during epigenetic change. The critical difference is that it's not a change to the identity of a nucleotide. So we've seen, for example, that CG dinucleotides, there can be methyl groups added to the cytosine, but that doesn't change its identity as a cytosine, and it would be read as a cytosine as, for example, transcription was occurring. Now, a similarity between the two is that they can be changed to produce gene expression differences. So both of these approaches, either mutations or epigenetic changes, can both affect gene expression. Quickly, an example of that might be if a mutation causes a change of DNA in the promoter of a gene such that it can no longer be transcribed, that would have an effect on gene expression. And we've talked a lot about how epigenetic changes cause chromatin structure changes, where DNA goes from euchromatin, where it's not highly compacted and it's easy to transcribe, to highly compacted DNA sequences where it's hard for proteins to get in there and undergo the processes that are necessary for transcription and translation. And that difference between euchromatin on top and heterochromatin on the bottom is often driven by epigenetic changes to histone proteins and to CpG dinucleotides.